Thank you very much, Hagan, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, I'm Hagan Bayley. I'm at the University of Oxford, and the work I'm going to talk about today was done in very close collaboration with Yuzhe Qing's group, also in the Department of Chemistry at Oxford. I changed the talk I was going to give because I got very interested in the protein content of cells, really quite indirectly by working on nanopore sequencing, and that's what I'll tell you about first. And then you'll see how we came up with, well, we were forced to think about the protein content of cells. And then at the end, it would be good if we could have a little discussion about the implications of what I'm going to talk about. So my slide didn't change. Let's try again. All right, let's try this one down here. There we go. All right. So we're interested in proteome analysis using nanopores. You're familiar with the DNA and RNA sequencing with the nanopores, which we'll come to briefly in a moment. But the story is that there are about 20,000 human genes that encode proteins, only 20,000 genes. And they make, I put 100,000 here, but they really make an unknown number of RNA transcripts that we still don't really understand how many there are and how they're modified and so on. And the RNA transcripts are now also really accessible by nanopore sequencing. But there are actually millions of different proteoforms from these 20,000 genes, and they come from familiar things like post-translational modification and RNA splicing and other phenomena that we'll come to very briefly in a moment. So, in other words, there's a huge amount of information in the proteome that can't be predicted directly from the genome. It must be in the genome somewhere, but it's not obvious how to pull this information out. And this information, being able to think about the proteome, is really important for both biology and medicine. And it occurred to me that it would also be very worth thinking about in terms of building synthetic cells. But our goal in terms of our work with nanopores is to be able to map post-translational modifications and splicing. And I would say just up front that we don't really consider it worthwhile on the whole to sequence polypeptides in this manner. It's going to be quite complicated and difficult to do and not provide much new information. But these post-translational modifications and so on are really vital. And our goal has been to map these within full-length polypeptides. So not just assemble statistics from fragments examined by mass spec, for example, but to put full-length polypeptides through nanopores and get actual populations of population distributions of modified forms. And in that way, we would be able to inventory all the various polypeptide species in cells, of which there are very, very many. And we would also aim to do this at the single cell level. And the conclusion I've come to recently is really that to do this at the single cell level is actually impossible. There's just too much information you would have to gather. And that's what I'll talk about today. And then we can discuss how that might apply to building a cell. So if you look at the Build a Cell manifesto, it actually does say we wish to create an organism for which all molecules are understood from a first order functional perspective. And I think that's actually going to be impossible, at least for complex cells that say along the lines of mammalian cells. So What are all these modifications? I mentioned post-translational modifications and splicing, but alterations to proteins can occur at the genome level. The genome itself can be altered, and of course they can occur at the RNA level through base editing as well as splicing. During translation, there are various different phenomena that will uh, change the amino acid that's inserted at a particular codon. And then in terms of direct protein modification, there are many, many different possibilities. Proteolysis, protein splicing, and reactive molecules that modify polypeptides and various chemical rearrangements and isomerizations. But just for now, I'm going to focus on post-translational modifications. So just to remind you about nanopore DNA sequencing, basically the bottom line is that company Oxford Nanopore has developed a device for threading single strands of DNA through a pore using a helicase nowadays. So the motion of the DNA is controlled enzymatically, and then it's not quite as easy as reading off one base at a time, but the sequence of the that's moving through the pore can be deduced from an electrical signal. And that electrical signal arises by applying a potential across the pore. Ions move through the pore, so there's an ionic current, and it's modulations of that current that allow you to read off a DNA sequence and nowadays an RNA sequence. So after a lot of work, more than 10 years ago now, the MinION was developed. This is a miniaturized DNA sequencing device that's really inexpensive and portable, can do ultra-long reads. They won't be quite so useful for proteins. And also there are, in the MinION, at least 500 pores working in parallel, so you can acquire a lot of information pretty quickly. So how might this be applied to proteins? Well, there aren't any enzymes that will shift amino acids one residue at a time into a pore. I think it might be possible to modify some existing enzymes to do that. And also, the polypeptides don't have a uniform, high uniform negative charge. So another important aspect of nanopore sequencing is to be able to pull on the DNA and stretch it while it's going through the pore. So first of all, we had to figure out a way to get polypeptides through pores and also unfold them at the same time. And we used a process called electroosmosis. And the way that works is illustrated here. You make a pore that has a high internal charge on the internal surface of the pore. This will make the pore ion selective. In this case, there's a positive charge, so the pore will become anion selective. And then when you apply a potential, say positive at the bottom of this pore, anions will move very rapidly through the pore, and they will carry a large amount of water with them, maybe up to 10 water molecules per anion. And that produces a so-called electroosmotic flow through the pore. And the force of that flow is strong enough to pull a polypeptide into the pore and indeed unfold a protein and have the entire polypeptide chain go through the pore and come out on the other side. So that's illustrated here and here. And we've been working mainly at the moment, this is very early days for this field. So we've been working mainly on model polypeptides, such as the one shown here, and trying to identify modified residues that we build into the protein. And that's not entirely the topic I'm chasing today. So let me just say in brief that we can modify these model peptides in various ways, phosphorylate them, put in short linear glycans, or attach peptides to them, various post-translational modifications that will occur in nature, and we can identify these modified residues as they pass through the pore. And as I said, we're looking, unlike sort of conventional mass spectroscopy, we're trying to look at full-length polypeptides. So illustrated at the top here is a polypeptide of over 1,200 amino acids, and we can identify a phosphoserine in the middle of this polypeptide, deeply buried inside the protein. And on the bottom is another one of about 700 amino acids, and we can identify two phosphorylated residues that are 274 amino acids apart. So we can do really long distance mapping of post-translational modifications. Now, one problem is that there are a lot of post-translational modifications. So more than 400 have been identified so far. Fortunately, maybe for us, there are about 13 abundant post-translational modifications, and they account for about 99.9% of the modifications that you will see in nature. Now, in the data I just showed you, or just went through very quickly, we were relying on current amplitudes, the extent of block of the ionic current by the post-translational modification, and other aspects of the signal, such as the noise in the signal, to identify the post-translational modification. But that has its limitations, and that's illustrated here for small molecule sensing with nanopores. The ideas, the concepts here are just also valid for biopolymers. But in general, we look at the duration of an event, but that's exponentially distributed, so it's very hard to identify a molecule or a base in a piece of DNA by the duration of an event. We can also look at the current amplitude, and that's illustrated on the right here. And you can ask yourself how many different current amplitudes might be identified by this approach. Maybe 10, maybe 20 at the very, very most. And then there are other information in these signals, such as the current noise, but that tends to actually reduce the separation of events, so reduces the number of different current amplitudes you can measure. So in other words, there are limitations to what you can do by just simply measuring the current as this protein passes through the pore. So that brought up this idea of using binders to assist in the discrimination between post-translational modifications. And I'll just show you quickly here one example where we have a long polypeptide chain that's phosphorylated. That's the pink signal here. And it also has a glutathione attached, a tripeptide. That's the blue signal here. And these have a very, very closely similar current amplitude. And one thing that we've been doing is looking for molecules that will specifically bind to these post-translational modifications. And illustrated on the bottom, it's actually real data, is a molecule called phostag, which was not invented by us. It was in the literature, but it binds phosphate monoesters very, very tightly. It's a sort of binuclear zinc complex. And you can see, again, just by looking at this trace at the bottom here, that the pink part of the trace is the phosphorylated residue when the phostag is bound. And you can see that looks very different to the phosphorylated part of the protein in the top trace. And you see this very sort of noisy signal. It's going between two different levels. And we believe that's because of transient breaking and reassociation of the zinc-oxygen bonds. But the bottom line is that it provides a distinctive signal for the phosphate group. And one thing that we're doing at the moment is trying to devise new binders that will look at this large variety of post-translational modifications. And there are two types that we're looking for, ones that will bind and go through the pore like the phos-tag, and others that will bind and be sheared off at the entrance to the pore. And the basic science of this is pretty interesting. We need 100% occupancy to be able to map these sites in polypeptide chains. These binders must work in water, so a lot of host guest chemistry that's done in dichloromethane won't work in water, obviously. And also these in the second type of binder, the one shown at the bottom, it needs to bind very tightly. It could be a nanobody or an aptamer, but we need to shear it off at the entrance to the pore, and we have to understand the effect of shear forces on the binding constants for these molecules. So that's where we're heading at the moment. The same ideas might be applied to RNA. There are a lot of different, these are just messenger RNA modifications here. And of course, what we'd like to do ultimately is have a machine that, like this tricorder on the left, that will do DNA, RNA, polypeptides, glycans, and small molecules, and be a little less complicated than the one that's was in Star Trek. So in the course of doing this, I realized there was a bit of a problem. And the problem is that there are 10 to the 9 to 10 to the 10 polypeptides in a typical human cell. And that means there are 100 to 1,000 times more peptide bonds than there are phosphodiesterase linkages in the genome of that cell. And if we could look at these polypeptide chains at 1 millisecond per residue, it would take about 2 months to analyze all the proteins in a single cell with a MinION device that had 1,000 nanopores working in parallel. So, in other words, this is a huge job compared to genome sequencing. And we'd either need a massive step forwards in sequencing technology that I'll mention briefly later, or we would need to enrich for our target proteins. So how many proteins are there in a cell? This is something that is still a little bit unsettled, but you can do a back of the envelope calculation if you assume that the protein content of a cell is about 100 mg per mil, and you make certain assumptions about the dimensions of the cell. In this particular, my back of my envelope, I came out with about 10 to the 9 molecules, protein molecules per cell. And then this really nice website, actually, with on bionumbers, there are various different estimates for different cells. And you can see they're really kind of all over the place, but they come up with 2 million for a mouse or a human cell. And then there are other examples in the literature as high as 10 million. So again, this sequence information greatly outnumbers the amount of sequence information in the genome. So the human genome is about 6 billion base pairs, and the protein content of a human cell, let's just call it 2 billion. And if we say there are on average 400 amino acids per protein, that we end up with about 800 billion amino acids, so roughly 800 billion peptide bonds. And a billion seconds is 32 years, so it would take more than a century to sequence all of these polypeptides. If you could use the MinION device with 500 pores, it would take about 93 days to do that and cost about $500. If you use the Prometheon, the sort of great sort of stack of MinIONs that's been put together by Oxford Nanopore, this will have almost 150,000 pores working in parallel. And you could do that now in 7 or 8 hours, but the running costs would be very high. So yeah, time is money, sort of well illustrated here. So we might ask then, so basically this is a really sort of impossible task. So that's a single cell. If you wanted to do 100 cells in a tumor or something, that would be very expensive or extremely time consuming. So if you want to look at rare proteins, that's another interesting question. So if you were looking at a single cell and you wanted a protein that had a copy number of 100 per cell, to identify and look at the modifications in those proteins, it would take you over a year to see a single copy and take you more than a century to see 100 copies. Again, that could be speeded up by using highly parallel devices. But even if you had 10 to the 6 copies of a protein in your cell, it may be one, we're looking at 1 in 1,000 or so, it would take you 111 hours, back of the envelope, to see 100 copies. And again, you can see the extent of the difficulty here. So another way to approach this from the nanopore point of view is to look at selected proteins. Now, one of the great virtues of genome sequencing, however it's done, nanopores or otherwise, is that it's a hypothesis-free technique where we don't ask to look at specific genes, we just sequence the entire genome. And if we wanted to look at selected proteins, the hypothesis-free nature of this approach would unfortunately be lost. Nonetheless, we could do that, say, by immunoprecipitation. And if after this enrichment, the protein were at the original concentration, given the way nanopore sequencing works, it's concentration dependent, the protein must diffuse to the pore, there would be no gain in analysis time. So you'd have to really concentrate the protein that you've immunoprecipitated or otherwise purified, and a 10 to the 6 copy per cell protein would need to be concentrated, say, 1,000 times. But if you did that and got all of those protein molecules into a microliter, the concentration would only be 1.7 picomolar, and you'd get very few events per second. And if we base it on the capture of DNA, and DNA is probably captured a lot more easily, you would see one event every 16 hours. So that would not be a technology really worth pursuing. You need to do something a bit clever to sequence these rare proteins. So just on the nanopore side, what we really would want is a sequencing technique that's 1,000 times faster. That's meant to be 1 microsecond per residue there. So it would take about a millisecond per polypeptide, and that would allow you to look at all the proteins in a single cell with 1 nanopore in just over a month. But if you used a MinION, you could do it in a couple of hours. So in other words, to get really comprehensive coverage with a nanopore of looking at protein modifications really requires a big step forwards in sequencing technology. I remind you that enzymes that handle DNA and the ones we know that handle proteins really work at about 1 or 2 milliseconds per residue. They don't work at a microsecond per residue, so it would require something drastically different. So after all that, I'll just come back to the topic, which is really a discussion topic. So from the builder cell, we would like to create organisms where we understand all the molecules in these organisms. And I would suggest what I just told you indicates that that's pretty well impossible. And if we think about that a little more, this is actually my last slide. As I said, in a mammalian cell, there are 2 billion or more proteins and at least 1 million proteoforms, probably many more than that. So how would you address that? Well, one way would be the way that people are going about things now to make the proteins and add them all at the start. And obviously, faced with this number of different proteoforms, that's impractical. And second, we don't even know what all these proteoforms are. Another approach would be to make your synthetic genome and jumpstart it in some way. So to put it into a giant vesicle and have all the goodies there that would get this genome working. And then obviously it's going to now make RNA and make proteins, and you just have to really hope it goes in the right direction. And point out there are really two types of complexity that's encoded for DNA, RNA, and polypeptides. And then we have a non-encoding encoded complexity in biopolymers, such as I've mentioned, polypeptide modification, but also for glycans as well. They are not made from a template. And if you're going to make a synthetic cell with no genome, the question is how ultimately could you generate the kind of complexity that you see in a mammalian cell with basically more than 1 million different types of protein in your synthetic cell. So I'll just end by thanking the people who we all went to Loch Ness, yes, in our group, and thank our funders. And I'll go back to the previous slide and ask if people think that this complexity is a worry in terms of build a cell, and if it is, how might it be addressed? So thank you.
Thank you so much. This was really awesome and kind of a sad for me. It's a downer that you think we won't be able to reach the complexity, but do you think we actually have to? There's some questions in chat, and I think we should move to those too, but I want to ask mine first. Do we actually have to make a minimal cell that's as complex as a natural cell, or can we get away with something much simpler that still does the functions of life?
Yeah, I think that's just the question, Kate, really. It's I'm just pointing out that it might well be impossible, A, to gain the knowledge of what's in a natural living cell, even to begin with. And even if we had that knowledge, it would be an absolute gargantuan task to put such a cell together. But then I think your comment follows, depending ultimately what you're trying to do. Are you trying to understand how a cell works? Are you trying to make a synthetic cell that will make a useful product? Yeah, I think that's also the very question I guess a lot of people in Build a Cell are addressing is, how simple a cell you can make that will carry out some useful or informative function. So I think, yeah, it's a good question. Now, I'm not sure how to get into the chat. You'll have to, all I see is
I
should,
could read you. I could read you the questions if
Yeah,
if you
go
prefer.
ahead. Or do I just have to escape from the presentation and then it will pop up somewhere? Or
I don't know. That depends on how your Zoom misbehaves. I'm happy to just read you the
yeah,
questions.
go ahead.
Okay, question from Jeremy. When you use nanopore to sequence PTMs, how do you deal with secondary structure like disulfide bridges that keep the protein folded?
Yeah, it's interesting that this is a much more difficult problem than sequencing DNA. And when we first started sequencing DNA, people thought we were mad even to take on DNA, which just essentially has 4 bases except methylation and so on, and they're all strung together in a linear fashion, no branching and whatever. So yeah, I think there are definitely issues. Disulfide bonds would have to be reduced and alkylated before you started, I think. Otherwise, you'd get these big loops that might not go through the nanopore. And then, obviously things like glycans are another problem. I mean, there are simple glycans attached to proteins, but there are very complicated branch ones as well. And I think the solution there, initially at least, will be to cut those off enzymatically, and at least then we can tell which residues were modified, even if we can't pin down the exact modification. But yeah, true. This is a very, very challenging problem.
Thank you. Next question from Everett. Do you think it's possible to estimate minimal number of proteins for a minimal cell, not complex living cell, but a tiny simplest one?
Well, I think this is the question many people in the field are addressing. I mean, you can see that there are certain single cells that, mycoplasma and so on, that just have about 50,000 proteins in the cell. I mean, they're so a single-celled organisms can work with less than 100,000 proteins in a cell. What the absolute minimum is, I think we don't know. But that must be getting close to it, I would guess. What would you think, Kate? Can you have a cell that really works, say, with just 1,000 proteins in? I'm not so sure.
Maybe 1,000 types of proteins. Probably not 1,000 pieces, but 1,000 different types. That's believable.
Yeah, that's right. I mean, I think you can obviously make a liposome of something that's got 1,000 copies of a identical protein in, and it will do something, but it's not a
Yeah.
synthetic cell.
Yeah. Okay. Next question from Eva. What about proteins made of multiple peptide chains? Is there a way to correlate information about how subunits go together after sequencing?
No, I think that's also a very good question. You see, I was very careful to use the word polypeptide rather than I slipped up every now and then. Actually, on this slide too, I've got proteins instead of polypeptides. So basically, the way we're doing it will break up any protein complexes, and we'll be looking at single polypeptide chains, and it won't be entirely obvious what the subunit composition of whatever complex they came from is. But there might be some clever way of pulling one polypeptide chain through a pore and then having its friends and relations hang out around the entrance to the pore and then pulling those in sequentially. I think that will be hard to do because they'll diffuse away pretty rapidly, but that's definitely another challenge, is to look at subunit composition of protein complexes. Yeah.
Mhm. Okay. Next question from Lucia. what is the accuracy of the system in identifying phosphorylation sites? Have you attempted to detect other post-translational modifications? And so how accurate were those detections?
Yeah, I think it's pretty early days. I think right now we're just showing that we can do it. So if we have phosphorylated sites in model proteins, we can detect them, but there are all various issues with electroosmosis and that the polypeptide chain is not moving at a constant speed through the pore and all sorts of things. So I think it's a bit early days to ask those questions. If I go back to the early days of DNA sequencing, we were just happy to identify a few bases, but then of course as time went on, we had to improve and improve and improve the accuracy of DNA sequencing and now for RNA. But I don't think we've really reached that point with polypeptides at the moment. So there's the question of, do we miss any? Do any just sort of go by and we don't see them? But there's also the possibility that I raised that two different modifications might give the same signal, and we're trying to get around that by using these binders, as I tried to explain. And then there's the other, third problem that some of these modifications are very similar to each other. So you can have a methylated lysine or a dimethylated lysine, and I think they're going to be very hard to distinguish. We'll probably know the lysine's modified, but to tell whether it's dimethyl or monomethyl will be quite hard. But as I said, it's pretty well early days yet. We're just trying to sort of work out the basic technology at this point.
Next question from Josh. I have a naive question. If you want to sequence a whole proteome, do we know what PTMs to expect? Do we know them all to know what current changes to expect? And what about some PTM we don't know about now? Would it be possible to identify unknown change in signal as new PTM?
Right. This is the kind of Donald Rumsfeld situation here with the unknown unknowns. Yeah, I think we know from sort of all the sort of mass spec that's been going on the last 20 years, I think we got a pretty good idea of the major modifications. And as I said, there are not really 13, some of those are glycans, so probably sort of 10 major modifications and we can go out and look for those. The rare ones, I think, are going to be pretty hard to pick up, but we will at least be able to say that that residue was modified in some way. And then are the completely unknown ones? That's we don't even really know that for DNA. DNA seems very simple. We just have methylated C bases, particularly in mammalian DNA, and then the oxidation products, the way they're broken down, sort of until they're cleaved off. RNA is more complex. There may be RNA modifications that have not been identified yet. So yeah, I think at this point we'd be just happy to look at really modifications that are, obviously really important, like phosphorylation, and take that as far as we can. But you're right, there'll be a lot of things still lurking there in the proteome that we don't know about yet.
Before we move on to the next question, I have my own follow-up. How many amino acids get post-translationally modified? Like if in a typical peptide of 100 amino acids, what percentage of them will be non-native?
I think that it varies a lot. So there are some cases, some protein kinases, for example, that have just activated by a single phosphorylation. And then there are other proteins like P53 or tau protein and so on that have many, many different, I'm just talking about phosphorylation now, phosphorylation sites, and they are, highly combinatorial, so that's one situation that gives rise to literally hundreds of different phosphorylation products from a single polypeptide chain. So I think there are, these so-called hyperphosphorylated proteins all the way down to proteins that may be regulated by a single phosphorylation.
Thank you. Okay, question from Paul. Would it be possible to make a minimal cell that does all functions of life but without PTM?
Yeah, that's also
That's a very qualified yeah.
yeah, I think that no, that just means I hadn't thought about it, right? It's a good question. Yeah, I think it depends what you wanted the cell to do, right? Obviously, if you wanted your synthetic cell to carry out some of the functions of a mammalian cell, where there's a lot of crosstalk between signaling pathways and a lot of things that are being turned on and off and so on, I think it might not be possible. So the thinking is, there are 20,000 proteins, but to get all these complicated things to happen in the cell, you need these modified forms, and also you need these modified forms to interact with each other. So phosphorylated proteins will be cleaved by phosphatases, for example. So there's a lot of interaction between all the different proteins in the cell. But could you make a simple organism? That's a good question. And I wonder, with these mycoplasma and so on that just have a very small number of proteins in the cell, whether there are actually less modifications, or maybe you need more to actually get the cell to work. Yeah, that was a copy number, the 50,000, not the number of different types.
Yeah, that's something that I was wondering myself, is with a tiny proteome, you have to multitask much more than with a big specialized one. So I wonder if modifications could be the way to increase the diversity from a small genome.
Yeah, exactly.
Okay, next question from Erin. Okay, she's asking really important question. Awesome talk. Did you find the monster?
We didn't find the monster, but you're a scientist too. You never give up, do you? So really, I think we have to go back, and definitely it's a very deep lake. We had sonar, we could see lots of big fish there, but there was actually no convincing evidence for the monster. And I suppose the monster could be the ultimate goal of Build a Cell, really.
Yes.
Actually, what I was going to talk about originally is our efforts to build tissues rather than cells, where the actual compartments interact with each other. But right now, we're just making a very small piece of tissue, but we, eventually go all the way and make the monster.
Yes, thanks for looking. Okay, Charles, thanks for the talk. Do you have any information on the geometry, for example, pore diameter, electrode placement of fabrication methods used in the nanopore systems?
Oh yeah, we basically invented that. So we have a lot of information. So really, we started with this protein called alpha hemolysin that you'll see in a lot of the pictures of nanopores, including our own. So that's a 7 subunit protein that's made by Staphylococcus. It's a toxin that will attack human cells, and that has the bottom part of the pore is a 7 subunit protein, 2 strands from each one, so it's a 14-stranded beta barrel, and that makes a channel of about 2 nanometers through the membrane. But there's it's that field itself is really fascinating. You know, there are small pores and large pores. There are huge pores. Streptolysin from Streptococcus forms a huge pore with literally a couple of hundred beta strands in the barrel. A common theme are these beta barrels that go through the membrane, although there are some alpha helical pores that are known as well. And then in our own bodies, you know, our own immune systems make proteins like the complement pore and perforin pore, so we can fight back against foreign invaders. But the whole field of protein pores, as opposed to ion channels, I mean, obviously ion channels is an amazing field as well. But by pores, I mean molecules that will let unhydrated ions through a membrane and will generally have a diameter of at least 1.5 nanometers, say. So they don't do single file transport.
Okay, thank you. Next question from Michael. Great talk as always, Hagan. As you said, people thought you were crazy to sequence DNA with nanopores, but you managed it. What do you think are the advances needed on the nanopore measurement techniques required to solve this for protein sequencing?
Yeah, I think interestingly, that's a lot of interesting philosophical questions here. You know, if you're sequencing a genome or a plasmid or a piece of DNA, you either can sequence it or you can't, right? It's really kind of all or none. I think on the protein side, we can find out interesting things about proteins even if we don't have the complete answer. You know, as one of the earlier questioners said, maybe we don't know all the modifications, but being able to map, say, 5 different modifications in a polypeptide chain could be very, very informative. So I think using what we know now, we will be able to make quite a bit of headway. But I think we need to explore new pores. I think ultimately we need to, by that I mean pores that have different diameters, different internal functionalization. I think we need to, ultimately, we're going to need to feed the DNA through the pore. I think the electroosmosis that I described is going to be a bit too unreliable, and that's probably going to require a lot of protein engineering on molecular motors that are already known to translocate polypeptide chains. They just don't do it in such an orderly way as helicases and polymerases and so on. Then I think that given the complexity of the problem, the parallel detection is going to be very important. So we would envisage having chips in flow cells that maybe contain 10, 20, 100 different types of pores, and they would be separately electrically addressable, so that we could get more information out of the system. Ultimately, I think that the present technology we have will be too expensive and too slow. So if someone can come up with a way of translocating biopolymers and identifying the monomers that constitute them at 1 second per base or amino acid rather than 1 millisecond per base or amino acid, obviously that would be a huge breakthrough as well. So I think there's a lot of work to be done. And I think in some ways, the basic science is just as interesting as the biology that's going to come out of it.
Thank you. I mean, I think that led to the partial answer to the last question from Cyril. Can you control the speed of peptide moving through the pore? Would slower improve accuracy?
Yeah, so going back, I keep going back to what we learned from DNA. Originally, we thought we could just put some DNA into our apparatus and apply a potential, and the DNA would move through, would be captured by the pore, and it's negatively charged, and it would be pulled into the pore and would move through it, and we could read off the sequence. So the problem there is it turns out that the DNA moves through a lot faster, at about 1 to 10 microseconds per base, than we can right now identify the bases. We can really only identify them on a sort of millisecond time scale. And that's where these enzymes came in for translocating the DNA, originally using polymerases, but later using a helicase. And they will work at a top speed of about 1 millisecond per base. But that's a controlled speed. With a helicase is ATP dependent enzyme, so you can control the speed by the amount of ATP that you have in the solution. So all that's kind of worked out for DNA, worked out to a certain extent for RNA, although that could definitely be improved, and really not worked out at all for polypeptides. So when you apply electroosmotic force, as well as pulling the polypeptide through the pore, it also unfolds the polypeptide chain that's outside the protein, although you can put denaturants into the solution and unfold them ahead of time. And then those polypeptide chains will move through the pore in a rather uncontrolled manner, or at least a manner we don't really understand at the moment. So getting control of polypeptide translocation would, yeah, indeed be very important, and we're not there yet on that one.
Thank you. Follow-up question from Charles. Is this a regularly spaced lattice of nanopores in a membrane for parallel sequencing systems? If so, how is the relative positioning controlled when assembling the membrane?
They're not actually. So basically, these chips consist of a large number of apertures that are spaced, a couple of hundred microns apart, and they will originally had a lipid bilayer, because these proteins, like these pores, like to insert into lipid bilayers. That's where they function. Nowadays, they contain a block copolymer, because that's more stable than a lipid bilayer, and you can then add your protein to, let's call it, the top solution, and if you add it at a low enough concentration, you will just get a Poisson distribution of pores in this array of membranes. So some will contain 0, some 1, some 2, and it's the ones that just contain one pore that you want. Subsequently, it was found out that you could control insertion with the potential applied to the membrane. It's not absolutely obvious how that works, but since in these chips, each aperture in the chip is separately electrically addressable, you can actually control insertion in that way, and then ensure that you just get one pore in each aperture. If you're using multiple pores, you would be able to apply them to the chip sequentially and record which aperture they go into, or you could have them insert into these apertures in a random way, and then really train the chip after insertion. So yeah, so it's important. And the reason you need them several hundred microns across is so you don't get any electrical crosstalk. We're measuring very, very tiny currents of 10 picoamps or 20 picoamps. So sort of any electrical interference of any sort is really has to be eliminated. Yeah.
Thank you. I think that's all the questions. Thank you so much.
Okay. Well, thank you, Kate. Thank everyone. And see you all soon.
See you soon, and hopefully I hope we will disappoint you and build a really complex cell where we'll understand everything, and then you'll
Well,
sequence the whole thing.
yeah. Or you make the monster, that
Yeah.
was suggested by one of your questions, right?
It's good to have goals.
Mm. Oh yeah, definitely so. And I think this is a very, this when we started looking at proteins, and it's good to be a bit naive because knowing the problems now, they're quite off-putting. But yeah, definitely good to have goals and lots of things still left to be solved in the world of science. Yeah.
True. All right. Have a great week, everyone.
All right. Bye. Bye. Thanks for
Bye.
listening.